To recognize and fight the wide range of pathogens an individual will encounter, the lymphocytes of the adaptive immune system have evolved to recognize a great variety of different antigens from bacteria, viruses, and other disease-causing organisms. The antigen recognition molecules of B cells are the immunoglobulins, or Ig. These proteins are produced by B cells in a vast range of antigen specificities, each B cell producing immunoglobulin of a single specificity. Membrane-bound immunoglobulin on the B cell surface serves as the cell's receptor for antigen and is known as the B cell receptor, BCR. Immunoglobulin of the same antigen specificity is secreted as antibody by terminally differentiated B cells, the plasma cells. The secretion of antibodies which bind pathogens or their toxic products in the extracellular spaces of the body is the main effector function of B cells in adaptive immunity. Antibodies were the first molecules involved in specific immune recognition to be characterized and are still the best understood. The antibody molecule has two separate functions. One is to bind specifically to molecules from the pathogen that elicited the immune response. The other is to recruit other cells and molecules to destroy the pathogen once the antibody is bound to it. For example, binding by antibody neutralizes viruses and marks pathogens for destruction by phagocytes and complement. These functions are structurally separated in the antibody molecule, one part of which specifically recognizes and binds to the pathogen or antigen, whereas the other engages different effector mechanisms. The antigen binding region varies extensively between antibody molecules and is thus known as the variable region or V region. The variability of antibody molecules allows each antibody to bind a different specific antigen, and the total repertoire of antibodies made by a single individual is large enough to ensure that virtually any structure can be recognized. The region of the antibody molecule that engages the effector functions of the immune system does not vary in the same way and is thus known as the constant region, or C region. It comes in 5 main forms, which are each specialized for activating different effector mechanisms. The membrane-bound B cell receptor does not have these effector functions, as the C region remains inserted in the membrane of the B cell. Its function is as a receptor that recognizes and binds antigen by the V regions exposed on the surface of the cell, thus transmitting a signal that causes B cell activation, leading to clonal expansion and specific antibody production. The antigen recognition molecules of T cells are made solely as membrane-bound proteins and only function to signal T cells for activation. These T cell receptors, TCRs, are related to immunoglobulins both in their protein structure, having both V and C regions, and in the genetic mechanisms that produces their great variability. However, the T cell receptor differs from the B cell receptor in an important way. It does not recognize and bind antigen directly, but instead recognizes short peptide fragments of pathogen protein antigens, which are bound to MHC molecules on the surfaces of other cells. Have a great day.